Steam rises in pale ribbons from the black mouth of the vent, curling upward into water so dark that light never arrives. The ocean here is vast and heavy, pressing from all directions, its weight more than any mountain's burden. Around the vent, minerals pour from the Earth's interior, iron, manganese sulfides drifting into the seawater, precipitating into delicate chimneys of brittle stone. The heat is immense, spilling from the planet's mantle in temperatures that would strip life apart in moments, yet here life begins. These early oceans are different from the seas of the later ages. The sky above them is veiled in a haze of volcanic gases, the sun's light dimmed and scattered by particulates. No oxygen yet drifts through the air or dissolves into the waves. The surface waters may glint faintly in scattered daylight, but far below near the mid-ocean ridges only the vent's mineral breath disturbs the darkness. It is in this darkness that the first Archaea appear. Their cells are simple to the eye without a nucleus or compartments, but each is a fortress against heat and pressure. They anchor to mineral surfaces where gradients of temperature and chemistry offer the energy they need. Instead of drawing light into sugars as plants will one day do, they turn the raw chemistry of the vent into living matter, capturing carbon from dissolved gases, building their proteins and lipids from the minerals around them. Their membranes are not the fragile bilayers of other microbes to come. They are built from isoprenoid chains bound together in ether linkages that do not break apart when the water boils. In these membranes lies one of their most quiet triumphs, the ability to remain whole where heat would unravel others. The plumes above them shimmer with metal particles like slow snow rising instead of falling. The heat that pours from the vent would melt ice instantly, but here in an ocean still in its geological youth it becomes a source of stability. Around the chimneys layers of minerals grow in concentric rings, each capturing a record of the vent's shifting chemistry. The Archaea weave themselves into this structure, forming thin films of life where rock meets water. In these films, individual cells divide slowly, not hurried by seasons or daylight. They measure time in the steady flow of minerals, in the rise and fall of temperature as the vent pulses with Earth's interior rhythms. This place is not silent. The water roars and hisses as it meets molten heat, yet it feels still in its constancy. There is an irony here, the very forces that seem most hostile to life, heat pressure, chemical volatility, are the ones that make it possible. The ocean's crushing depth shields these microbes from the ultraviolet radiation that scours the surface. The heat that would kill surface organisms stabilizes the enzymes of these deep dwellers. The mineral-laden water, toxic to later life, is their nourishment. Another irony is that life here has no need for the sky. The sun's great energy, the star that will later drive forests and reefs, is irrelevant in these depths. The Archaea thrive in a realm entirely decoupled from sunlight, making a mockery of the idea that life must always follow the light. And perhaps the most enduring irony is that these beginnings happen without witnesses in darkness so complete that they will leave no direct trace for billions of years, save for chemical whispers locked in ancient stone. Scientists still do not know the exact sequence of steps by which such cells emerged. Did they arise here in vents like this, where mineral surfaces could act as catalysts, concentrating and assembling the molecules of life? Or did they first evolve elsewhere in shallower pools, or even beneath the crust itself, and then spread into these deep hot habitats? The rocks hold clues, but they speak slowly and not always clearly. Some isotopic ratios hint at early microbial activity. Certain mineral formations suggest biological mediation. Yet the full picture remains incomplete, fractured by time and geology. Modern vent systems give hints, but they are not identical to those of the early Earth. The ocean's chemistry has shifted, the crust has aged, and oxygen now seeps into places it once never reached. Still, these ancient analogues teach us that life's limits are not fixed at the comfortable middle, but stretch into extremes once thought empty. The vent's plume continues to rise, a column of heat, in a world without continents, without green. Above it, currents carry dissolved minerals outward, where they will settle into the abyssal plains, adding their weight to the slow architecture of the seafloor. The Archaea continue their quiet labour, each cell a small workshop of chemistry, indifferent to the cold darkness beyond the vent's reach. This place will change over time, chimneys will collapse, new ones will form, flows will shift, but for now it is a constant refuge. Here, under unimaginable pressure and heat that blurs the boundary between water and steam, life's first fortresses stand unshaken. They do not move toward complexity for its own sake. They do not need to. Their strength is in remaining in holding their form while the planet's surface writhes and churns above them. And in that persistence they will shape the course of Earth's future, though they cannot know it. The salt flats stretch outward under a sky that is not yet the blue we know. Instead, it carries a muted tint veiled with fine dust and volcanic haze. The sun dimmed to a pale disk above the horizon. The air is thin, the wind slow, and the surface shimmers as though glazed with glass. Here shallow pools have evaporated under the young sun's unfiltered light, leaving behind vast sheets of crystallized salt. Each grain catches what little sunlight there is, scattering it into a brightness that belies the stillness of the place. Beneath the crust, brine remains dense hypersaline water, trapped between layers of crystalline halite. In this concentrated solution, few forms of life can endure. Osmotic pressure draws water out of any unadapted cell, collapsing it into nothing. Yet in this apparent sterility, Archaea thrive. Halophils, they are called salt lovers. They do not merely endure the saline environment, they require it. Their proteins, their enzymes, their very structure is tuned to the high ionic strength of these brines. Remove them to fresh water, and they would rupture in reverse, overwhelmed by the imbalance. Their membranes, rich with specialized lipids, form an unyielding barrier to uncontrolled water loss, while their proteins are studded with acidic residues that remain stable in salt-saturated conditions. In the most concentrated pockets of brine, where salt begins to precipitate out in fine cubes, the cells cluster into thin films, their pigments tinting the liquid with subtle hues, reds, oranges, sometimes violet. These pigments are not for photosynthesis in the plant sense. Instead, they often function as shields, absorbing the harmful ultraviolet radiation that slips through the atmosphere. Some halophils, however, make use of light in another way through bacteriorhodopsin, a protein that uses sunlight to pump protons across a membrane, generating energy, in the absence of traditional photosynthetic machinery. It is a quiet irony that these cells living in the blinding glare of reflected sunlight are not undone by it, but instead bend it to their advantage. Another irony lies in the nature of their home salt, which preserves and sterilizes in the human world. Here forms the very foundation of an ecosystem. The salt crust is both shield and stage, shielding the cells from the shifting winds, and staging their growth in thin films where nutrients are concentrated. A third irony is that while these flats appear lifeless from a distance, their colours, textures, and micropatterns tell a story of dense microbial occupation hidden from the casual glance. Scientists can only piece together fragments of how such halophils arose. Did they emerge first in inland seas that evaporated under early heat, or in marine bays cut off from the open ocean, concentrating their salinity over centuries? The geological record is sparse. Ancient evaporates are rare and often transformed by later tectonics. Modern analogues like the Dead Sea or Lake Retba offer insight but cannot fully reconstruct the ancient conditions. What is certain is that these environments have existed for most of Earth's history, forming whenever evaporation outpaces inflow, and Archaea have been present to claim them. The flats themselves change slowly. layers of salt build over decades and centuries, each recording a season's evaporation, each layer trapping air bubbles and organic traces. In cross-section, these layers become a stratified archive, whispering of past climates of shifting chemistry. The halophiles drift through these layers over time, trapped in crystals, surviving in a dormant state, until the salt dissolves once more. In this way, the salt flats are not just a home, but a long-term vault for life-preserving cells across centuries. Under the muted sky, the horizon trembles in heat haze. The reflected sunlight makes the surface appear as water from a distance, a mirage that draws the eye. This illusion is another quiet reminder appearances here are deceptive, and what seems inhospitable can be rich with life. The wind is soft, carrying only the faint hiss of crystals shifting under their own weight. No waves break, no leaves stir, no animal footprints cross the surface. Yet within the brine pockets, chemical reactions hum at a molecular scale unchanged for eons. The halophiles replicate slowly, not pressed by seasons or predators, their pace determined instead by the slow dissolution of minerals and the arrival of occasional rain. When the rare storm sweeps in, the surface floods, the crystals dissolve, and the brine becomes briefly more dilute. For most organisms this would be relief. For the halophiles, it is disruption. They are just altering the flow of ions across their membranes, switching metabolic pathways to match the new balance, waiting patiently for evaporation to return their home to its preferred saturation. The water recedes, the salt blooms again, and the cycle resumes. In the deep time of Earth's history, such places will come and go, forming on different continents, in different climates, under different skies. But the lineage of the halophiles will remain, moving from basin to basin, carried in dust storms, in migrating seabirds' guts, in droplets borne by the wind. The salt flats are quiet now, as they were in the beginning. The sun sinks lower, its reflection stretching across the mirrored surface. Pigmented films just beneath the crust absorb the last light, their molecules shifting in response, pumping ions, storing energy. Above them the air cools slightly, and the first thin crystals of nighttime frost begin to form in the driest corners. The halophiles continue undisturbed. They have no need for change, for movement, for novelty. Their survival depends on sameness, on the repeated dance of evaporation and crystallization. And in that constancy they persist, holding their place in a world that will, over the next billions of years, transform almost beyond recognition. The chimneys rise like blackened spires from the seafloor, their surfaces rough with mineral deposits, their hollows coursing with water hot enough to scald on contact. This is a place of shifting gradients, heat and cold, acidity and alkalinity, chemical, richness, and scarcity, all meeting in narrow bands where life can take hold. Around the base, plumes of superheated fluid billow upward, charged with hydrogen sulfide methane, and metals drawn from the mantle. The water here is not simply warm, it ranges from near freezing in the surrounding abyss to more than 350 degrees Celsius within the vents themselves, though it does not boil under the crushing weight of the deep ocean. It is in these mineral labyrinths that Archaea first find the stability they need to endure. They adhere to the vent walls, building biofilms that ripple in the rising currents, their cells locked together in a mesh of extracellular polymers. Each vent is like a fortress of stone built grain by grain from the mixing of vent fluid and seawater, and within these walls temperature and chemistry shift in gradients, fine enough to be navigated by microscopic life. The Archaea use these gradients to their advantage. Some harvest chemical energy from hydrogen or methane, oxidizing them to build organic molecules. Others specialize in sulfur metabolism, reducing or oxidizing elemental sulfur, depending on the surrounding chemistry. The diversity is subtle, invisible without magnification, but it reflects the adaptability of these early cells. In the darkness, without the need for light, they flourish in a self-contained world. One irony of these systems is that their heat, which seems to offer boundless energy, is also their greatest instability. Chimneys collapse under their own weight, or are buried by mineral flow, forcing their inhabitants to disperse and recolonize. Another irony lies in the fact that the vents are both ephemeral and ancient. Each individual structure may last only decades, yet the process that forms them has been continuous for billions of years. A third irony is that the minerals, which in other contexts become inert rock, here serve as scaffolds for life, holding it steady against the relentless current. The exact path by which Archaea colonized these vents is unknown. Some researchers suggest they originated here, their biochemical adaptations forged in the interplay of heat and chemistry. Others imagine that they arrive from more temperate environments drawn by the steady supply of chemical energy. The rock record offers only indirect hints, unusual isotopic signatures in ancient sulfides or microstructures that may be fossilized microbial mats, but these traces are ambiguous, easily confused with abiotic mineral patterns. Modern vents, though younger and chemically altered by the oxygenated ocean, still serve as living laboratories for these questions. The chimneys grow layer by layer, depositing concentric shells of sulfide minerals. Within these layers, microbial communities adapt to the shifting chemistry, their metabolic networks weaving through the rock's microscopic pores. Some cells remain anchored deep within the vent in darkness and constant heat. Others occupy the outer surfaces, enduring cooler temperatures and more turbulent flow. This stratification mirrors the gradients of the early Earth's vents, offering a map of where different lineages might have thrived. The water around the chimneys is never still. Superheated fluid meets cold seawater in a roiling interface that precipitates fine mineral particles, creating a shimmering halo of suspended matter. Within this halo, Archaea drift as free-living cells or in small clusters carried upward before settling again onto rock or being swept away into the abyss. The life here is invisible to the unaided eye, yet its presence alters the chemistry of the vent. Hydrogen sulfide is consumed, methane is generated or oxidized, elemental sulfur is deposited in soft yellow filaments. These changes may seem slight, but over geological time they influence the composition of the oceans themselves. The vents are more than habitats. They are chemical engines cycling elements between the crust and the sea. Above them the ocean is vast, but the Archaea do not spread far into the open water. Their adaptations are precise, tuned to the mineral-rich vent fluids, and their survival depends on the constancy of this environment. Yet when vents shift or collapse, cells are inevitably carried away, settling in distant cracks or depressions, sometimes surviving, sometimes not. This slow dispersal may be how such life spread across the planet from ridge to ridge, basin to basin. The geological persistence of hydrothermal activity ensures that there is always another refuge somewhere. The mystery that remains is whether these Archaea are relics of a singular origin or the product of multiple independent adaptations to vent life. Genetic studies reveal deep evolutionary branches suggesting a complexity in early microbial history that is still not fully resolved. Perhaps vents have been both cradle and refuge places where life began, and places where it retreated during global upheavals. In the present, the chimneys stand silent to human ears, but their plumes are filled with the hiss and turbulence of mixing waters. The Archaea do not sense the structure's fragility. They inhabit it as though it were eternal. Mineral particles settle onto their biofilms, embedding them in a growing crust that may one day entomb them entirely. Even then, some will survive dormant within the rock until heat and chemistry align once more to awaken them. Around the base, the seafloor is carpeted with mineral sediment, the product of centuries of vent activity. Here, away from the hottest flows, conditions are stable enough for slower growing species, those that endure lower temperatures and nutrient trickles. These fringes may act as stepping stones between vents, allowing cells to persist between eruptions of activity. In the unchanging dark, time feels suspended. The chimneys breathe their mineral plumes into the abyss just as others did long before and as others will after. For the Archaea, each chimney is both beginning and end, a temporary home within a process that spans the planet's entire history. In water so hot it wavers between liquid and vapour, most cellular membranes would fail in moments. The delicate bilayers of bacteria and eukaryotes built from fatty acids joined by ester bonds begin to lose integrity as heat accelerates the motion of their molecules. Gaps form, permeability rises, and the precise chemical gradients on which life depends are lost. Yet the archaea that inhabit these boiling springs and deep-sea vents hold their form without strain. Their membranes are not made like others. Instead of fatty acids, they use isoprenoid chains, long and branched, tethered to glycerol backbones by ether bonds that resist hydrolysis. Some even link the two halves of their membrane into a single continuous sheet, a monolayer that cannot peel apart under stress. This molecular architecture is more than an adaptation. It is a declaration of independence from the fragility of most life. In environments where heat unravels proteins and disrupts every other known form of membrane, the archaeal cell envelope remains steadfast, sealing the interior against both thermal and chemical assault. The lipids are densely packed, their branches interlocking to reduce fluidity, so that even at temperatures above 100 degrees Celsius, the membrane holds the consistency of a cool gel. Inside, enzymes function with equal precision, their folding stabilized by networks of ionic bonds and hydrophobic cores that resist denaturation. There is a quiet irony in this strength. The very rigidity that grants survival in extremes would be a hindrance in the mild environments most organisms occupy. These membranes are overbuilt for the ordinary, locking their owners into a narrow but secure ecological niche. Another irony lies in the fact that such ancient molecular solutions remain unmatched by later life. After billions of years of evolution, no eukaryote has developed anything comparable in thermal endurance. And perhaps the subtlest irony is that these adaptations may have arisen not in response to heat alone, but as a byproduct of early chemistry only later becoming essential in hot environments. The origins of these membranes remain a point of uncertainty. Some researchers argue that they reflect a very early divergence between the archaeal and bacterial domains, with each inheriting a different set of building blocks from the last universal common ancestor. Others suggest that the archaeal architecture evolved independently, shaped by selection in extreme habitats. Modern biochemistry can compare the pathways and precursors, but the rocks hold few direct clues. Lipids degrade over geological timescales, leaving only rare chemical fossils that must be interpreted cautiously. In modern hot springs, the resilience of archaeal membranes can be seen in action. Cells cling to the edges of scalding outflows in water that shifts from near boiling to cooler trickles over just a few centimeters. In this steep gradient, other microbes find refuge in cooler zones, but archaea occupy the very limit, forming dense mats where temperatures hover near their upper threshold. Even here, their internal chemistry remains controlled, their membranes unmoved by the heat that distorts the structures of their neighbours. The uniformity of these membranes across diverse lineages suggests an early and decisive commitment to this design. While bacteria and eukaryotes have endlessly varied their fatty acid tails, adding unsaturations, altering lengths and branching patterns, the archaea have held to their isoprenoid ether chains with minimal change. This stability may have allowed them to colonize new extreme environments quickly, without needing to reinvent the core of their cellular envelope each time. Yet the membranes do not work alone. They are part of a larger suite of adaptations, specialized chaperone proteins, that refold heat-damaged enzymes, solutes that stabilize intracellular structures, DNA-binding proteins that protect the genome from thermal breakdown. Each layer of protection supports the others, making the whole system more resilient than the sum of its parts. In deep time, these membranes may have given Archaea an advantage not just in survival, but in persistence through planetary upheavals. Global impact, volcanic winters, and fluctuations in ocean chemistry would have erased many habitats, but the hot springs and hydrothermal systems where these cells thrived were among the most stable environments on Earth. Shielded from surface chaos, Archaea could continue their quiet existence, passing down the molecular blueprints for their indestructible membranes through unbroken lineages. Standing at the edge of a modern vent field or thermal pool, one might see the surface shimmer with oily films or ripple with rising gas. Beneath archaeal cells drift and settle their membranes, flexing minutely with the movement of ions but never losing cohesion. The water may be cloudy with mineral precipitates, yet each cell maintains a precise boundary between itself and the world outside. In this they embody one of life's most fundamental principles, the creation and maintenance of a self within an environment that constantly seeks to dissolve it. The membrane is both barrier and bridge, keeping the outside out while letting the necessary flow of matter and energy pass through in controlled measure. The chemistry inside an archaeal cell is as ancient as the membrane that contains it, a balance of reactions tuned to an environment that would annihilate most other life in seconds. In their stability lies the foundation for all their other adaptations, for without this unwavering boundary, no metabolic pathway, however ingenious, could function here. It is a truth that has endured since the first of their kind sealed itself off from the chaos around it. Survival begins at the membrane, and in the heat that melts other worlds, these walls still hold. Far from sunlight, where the ocean presses with unyielding weight, and the cold is broken only by isolated pulses of heat. There are forms of life that draw their sustenance not from plants or photosynthesis, but from the raw chemistry of the earth itself. These archaea breathe without air, eat without plants, and live without light, transforming metals and gases into the molecules they need. Some take electrons from hydrogen or iron, others from sulfur compounds, channeling them into metabolic pathways that build organic matter from dissolved carbon dioxide or methane. In these lightless depths, oxidation and reduction replace the familiar cycles of day and night. Here an irony is immediate. The energy that drives them comes not from the sky, but from the stones beneath their anchorage, a reversal of the pattern that will come to dominate most of Earth's surface life. Another irony is that what we think of as toxic hydrogen sulfide, ferrous iron, or methane in high concentrations is for these cells as vital as oxygen is to humans. A third irony lies in the very invisibility of their work. Though they may exist only in microscopic films, clinging to rocks or drifting in plumes, their chemical transformations subtly alter entire ecosystems, influencing the composition of the water for kilometers around. The mechanics of their metabolism are as precise as they are ancient. Sulfur-reducing archaea, for instance, take elemental sulfur and convert it to hydrogen sulfide, a reaction that both yields energy and feeds into further chemical loops involving bacteria and the mineral substrate. Methanogens, among the most primitive seeming of all archaeal lineages, combine carbon dioxide with hydrogen to produce methane, a simple molecule, yet one that plays an outsized role in climate, both in the present and in the distant past. This methane exhaled invisibly into the deep rises slowly through the water column. In the early Earth's atmosphere, before oxygen took hold, it may have been a crucial greenhouse gas trapping heat from the faint young sun and keeping the planet's surface from freezing solid. Modern research supports this view, but the details of how much methane was produced and by which communities remain uncertain. The rock record is silent on flux rates, and the isotopic fingerprints are faint after billions of years of alteration. What is clear is that these metabolisms are not adaptations to a rare set of conditions, but are deeply embedded in the planet's geochemical cycles. Iron-oxidizing Archaea, for example, can grow at the interface between anoxic vent fluids, rich in Fe2+, and more oxidized seawater, precipitating rust-colored films of iron oxides in their wake. These precipitates become part of the sediment record-forming layers that will later harden into banded iron formations. Such processes blur the line between biology and geology, making it difficult to say where one ends and the other begins. The energy available from these reactions is modest, far less than that provided by sunlight to plants or cyanobacteria, and so the growth of these communities is slow. A single cell may divide only once every few days or even weeks, an almost unthinkable pace compared to the rapid cycles of surface microbes. Yet this slowness is part of their stability. In the absence of predators or competition for light, there is no evolutionary pressure to outpace neighbours, only the quiet persistence of chemical equilibrium. In the modern world, scientists seek these organisms in deep subsurface boreholes, in hydrothermal vent plumes in cold seeps where methane seeps upward from buried sediments, and in hypersaline or acidic pools where metals are abundant. Each new environment offers a slightly different chemical palette, yet the same underlying principle holds. Life can be sustained wherever there is a gradient to be exploited. The molecular machinery that allows this enzymes, specialized to bind metals, electron carriers stable under extremes membranes impermeable to ions, except through controlled channels, is a suite of adaptations as intricate as any found in the more familiar forms of life above. The mystery is not so much how they function now, but how such pathways arose in the first place. Did the earliest Archaea inherit them from an even older ancestor that lived in the pores of mineral chimneys, using primitive catalysts formed on iron-sulfur surfaces? Or did they evolve later as cells spread into new niches and found novel chemical gradients to tap? Without direct fossils, the answer remains hidden, and the debate continues. Still, the chemistry of the modern deep ocean offers a clue. Many of the compounds Archaea metabolize are also produced abiotically in hydrothermal systems, suggesting that life may have arisen in close partnership with the geochemical processes that continue to shape such habitats today. In this view, the Archaea are not merely adapted to vents and seeps, they are continuations of the very mineral reactions that predate biology, carried forward in a living form. In the dark, a vent field exhales its plumes, the metal-rich water curling and folding in the stillness of the abyss. Within it, clusters of Archaea drift settle, and begin the slow work of transforming invisible molecules into the matter of life. They leave no visible mark beyond the subtle staining of minerals, yet their presence changes the balance of gases, the form of elements, and the texture of the sediments. In a world without light, they are architects of invisible landscapes. This is not a temporary role. For billions of years, as continents drifted and seas opened and closed, as climates shifted from ice to heat, the deep habitats where these cells endure have remained. Here, far below the turbulence of weather and sunlight, the same quiet metabolisms continue. Their constancy has shaped the chemistry of oceans and atmosphere alike, long before the rise of oxygen-breathing animals, and will continue long after many current life forms vanish. In the slow cycles of the deep, where centuries pass unnoticed, the Archaea breathe metal and stone, and in doing so bind the living world to the planet that sustains it. In the blinding whiteness of hypersaline ponds, where the air itself seems thick with reflected light, the water holds more salt than most life can bear. The brine is dense, its surface sluggish to ripple in the wind, and every drop is saturated with crystals so fine they remain suspended, refracting the sun into a haze. Here, halophilic Archaea are not simply present, they dominate. The water glows faintly with their pigments, especially when seen from above deep reds and magentas that spread across the surface in irregular patches, the work of carotenoids and retinal-based proteins. These colours are more than aesthetic. Carotenoids protect against the ultraviolet light that easily penetrates the thin atmosphere of early Earth, while proteins like bacteriorhodopsin capture sunlight in a primitive form of phototrophy, pumping protons across membranes to store energy. The irony is that these organisms live in conditions so harsh that most phototrophs would die instantly, yet they still harness light in a way that sidesteps the elaborate machinery of chlorophyll-based photosynthesis. Another irony is that their habitat so hostile to most microbes is open to evaporation and temperature swings that would seem to threaten stability, yet it remains one of the most predictable ecosystems on Earth. Salinity here is always high, and nutrients arrive in slow, steady trickles from dust rain or mineral seepage. A third irony lies in their pigments. What is a protective measure for the Archaea becomes from above a visual flag, announcing the very habitat that is meant to be inhospitable. The mechanics of their survival are embedded in every part of their physiology. Their proteins are rich in acidic residues, giving them a negative charge that attracts a surrounding cloud of water molecules, counteracting the dehydrating pull of the salt-rich environment. Their DNA is stabilized by high concentrations of potassium ions inside the cell, matching the osmotic pressure outside and preventing catastrophic water loss. Even their ribosomes, the molecular machines of protein synthesis, are tuned to function in ionic conditions that would instantly halt the machinery of bacteria or eukaryotes. In these environments, competition is rare. Few other lineages can withstand such osmotic extremes. As a result, halophils often grow in dense, nearly uniform populations, forming layers of cells just beneath the water's surface, where light is abundant but exposure to damaging radiation is limited. The calm surface belies the microscopic complexity below, where gradients of light, oxygen, and nutrient availability shape the arrangement of microbial mats. In the shallows, where evaporation is fastest, the salt crust thickens, and cells may become trapped in the growing crystals. Some remain viable for years in this suspended state, reviving only when the salt is dissolved by rain or flood. This capacity for long-term dormancy is both a survival mechanism and a form of dispersal. Windblown salt can carry living cells across continents, seeding new hypersaline lakes and coastal flats far from their origin. The question of how halophilic Archaea first arose is unresolved. Some researchers suggest they may have evolved from ancestors in marine environments that gradually became isolated and concentrated by evaporation, adapting step by step to the increasing salinity. Others imagine a more abrupt transition, with lineages moving directly from moderate to extreme habitats forced by environmental collapse or competition. Geological evidence is fragmentary salt deposits do not always preserve organic matter well, yet chemical traces in ancient evaporates suggest that halophiles have been part of Earth's biosphere for at least hundreds of millions of years, and perhaps far longer. In modern settings, such as the Great Salt Lake, Lake Magadi, or solar evaporation ponds, halophilic Archaea still mirror these ancient strategies. They form phototrophic surface layers where bacteriorhodopsin works alongside other light-driven proton pumps, converting photons directly into a usable electrochemical gradient. Unlike plants, they do not fix carbon through complex cycles. Instead, they use stored energy to support simpler anabolic reactions, an approach that is efficient under nutrient-poor high-salt conditions. The communities that result are self-sustaining recycling organic matter within their mats and sediments with minimal dependence on external inputs. When the light fades in evening, bacteriorhodopsin-driven pumping ceases, but the cells remain stable, their membranes and proteins unaffected by cooling temperatures. At night, under a sky unpolluted by modern haze, the stars would reflect in the salt-crusted water, fractured into a million points by the rippled surface. In such darkness, the halophils persist as they have for eons, indifferent to the passing of days. The next morning, as the first light strikes the surface, the pigments again capture what they need, and the slow work of growth resumes. Over geological timescales, such habitats appear and vanish. Inland seas shrink, tidal flats rise and fall with tectonic shifts, and climates swing between wet and arid. Yet halophiles remain ready to colonize each new salt-rich basin. This readiness is partly due to their dormancy in salt crystals, but also to their genetic stability. The very conditions that slow their growth also slow the accumulation of mutations, giving them a remarkable continuity across ages. If a pool dries completely, leaving behind only salt, the wind will eventually carry that salt and the life within it to another basin. In this way, the lineage is not confined to a single location, but follows the planet's shifting hydrology like a shadow. The persistence of halophales offers a clue for astrobiology if life exists, or once existed on other worlds with evaporative basins. Mars, for example, it may have left traces in salt deposits that endure long after the water is gone. Salt, it seems, is not just a challenge for life, but also one of its best preservation media. The irony here is deep. The same crystals that in human history have been used to kill microbes and preserve food serve as both shelter and vehicle for these Archaea. And so beneath the blazing sun of the early Earth in water, too saline for almost any other form of life, they spread their pigments like a quiet banner over the surface, marking their place in a world that is still learning how to host complexity. They will remain long after seas change their shapes and climates turn their resilience, built into every molecule of their being, each cell a memory of the salt from which it came. The air above the pool carries a metallic tang, and the surface shimmers with colours that shift from ochre to emerald to milky white depending on the minerals dissolved below. Here the water is not only hot but strongly acidic, with a pH so low it can etch metal and bleach the skin from bone. Steam rises in slow curls, carrying the faint sting of sulfur dioxide and other volcanic gases. These pools are fed from deep within the crust where groundwater meets magma-heated rock, and emerges infused with acids formed from volcanic emissions. For most life, such waters are instant death, the proton concentration alone enough to unravel proteins and dissolve cell walls. Yet in these conditions, acidophilic Archaea live and grow. Their membranes, already distinct from those of other life, are further modified to resist proton invasion, with tightly packed lipids and protein channels designed to control the flow of ions with exquisite precision. Inside their cytoplasm maintains a near-neutral pH, even as the environment outside is thousands of times more acidic. The proteins they use for metabolism are unusually stable, their structures reinforced by additional ionic bonds and compact folding patterns. An irony lies in their choice of home. They inhabit places so chemically aggressive that even the minerals in the surrounding rock are in a constant state of dissolution, yet their molecular architecture remains intact for years. Another irony is that these organisms can in some cases accelerate the very corrosion that would destroy them if they were unprotected, as their metabolic processes release more acid into their environment. A third irony is that while such habitats appear unstable, shifting, depositing new minerals, their chemical composition can remain remarkably consistent over time, offering a predictable stage for life finely tuned to its extremes. The exact evolutionary steps that led to acidophily remain uncertain. One hypothesis is that the earliest acidophils evolved from organisms in neutral hot springs, gradually adapting to increasing acidity as volcanic emissions intensified, or as water became trapped in basins with high sulfur input. Another possibility is that life began in acidic environments from the outset, making neutral pH adaptation the exceptional case. Evidence from modern acidophiles suggests that their tolerance is not an afterthought, but an integrated feature involving changes not only to membrane composition, but also to enzyme surface chemistry, DNA-binding proteins, and even the folding of ribosomal RNA. In places like the Dalol hydrothermal field in Ethiopia, or the Rio Tinto in Spain, these adaptations are visible in vivid form. Microbial mats grow on the edges of acid pools, forming rust-coloured biofilms fed by dissolved iron and sulfur. In some cases, acidophilic archaea partner with bacteria in cooperative cycles. One oxidizes iron to gain energy, producing acidic ferric ions, while the other reduces the iron again, recycling both the metal and the acidity. Such interactions create self-sustaining micro-ecosystems in conditions that would seem incapable of supporting even a single species. The colours of these pools are not just mineral deposits but microbial signatures. Yellow sulfur crusts are laced with colonies of sulfur-metabolizing archaea. Green films may be rich in iron oxidizers. In the early Earth, before the oxygenation of the atmosphere, acidic pools may have been far more common, forming wherever volcanic gases met standing water. The modern analogues provide a living window into these ancient conditions, though the chemistry has shifted over time with changes in atmospheric composition. The persistence of acidophiles offers a quiet reminder: what appears destructive to one form of life can be the foundation for another. The proton gradients that corrode rock and metal also provide a ready source of energy for cells capable of capturing it. By allowing protons to enter the cell only through controlled channels linked to ATP synthase acidophiles, turn environmental hostility into metabolic advantage. This use of natural gradients is one of life's oldest strategies, and here it is played out in its most concentrated form. The landscapes they inhabit change on multiple timescales. Over days or weeks, mineral crusts form and collapse, redirecting flows and altering the shape of pools. Over centuries, volcanic activity shifts, creating new acidic springs and extinguishing old ones. Yet wherever new acid pools appear, archaea are among the first to colonize them, carried by wind, water, or animals. Encysted cells dormant within mineral grains awaken when conditions align, beginning the cycle anew. As steam drifts from the water's surface, droplets condense and fall back in slow arcs, carrying with them both dissolved acids and microscopic life. In the stillness of early morning, when the air is cool and the steam rises more sharply, the pool seems almost lifeless. Yet under magnification, every surface is busy ions, moving across membranes, enzymes folding and refolding, protons slipping through molecular gates, each event invisible on the human scale but essential to the cell's survival. The paradox is that such places are at once dynamic and constant. The physical features shift, yet the chemistry remains within the narrow range to which these organisms are adapted, and so they persist untroubled by the volatility that would make other ecosystems fragile. The role of acidophiles in planetary history may extend beyond Earth. On worlds with volcanic activity and surface water, acidic springs and lakes could offer the same combination of energy gradients and mineral interfaces that make them habitable here. In the search for extraterrestrial life, the signature of acid-loving metabolisms, certain mineral assemblages, distinctive isotopic ratios could be as telling as the more familiar signs of photosynthesis. Standing at the edge of an acidic pool, whether on the modern Earth or in the imagination of another planet, the contrast is stark. The stillness of the surface against the chemical violence, beneath the dissolution of stone against the persistence of life. The archaea that endure here are not merely survivors. They are opportunists of a kind so specialized that they have no need to move beyond their narrow world, and in that narrowness they find stability thriving in waters that erase almost everything else they touch. Far below the reach of light, deeper even than the most remote hydrothermal vents, Water seeps through fractures in the rock, threading its way through pores and fissures in the planet's crust. Here, kilometers beneath the surface, the weight of overlying rock compresses every void. Yet space remains for thin films of water clinging to mineral grains or coursing slowly along fault lines. Temperatures vary with depth, sometimes near freezing where water has percolated from the surface, sometimes exceeding 100 degrees Celsius where geothermal gradients run steep. In this deep rock biosphere, Archaea persist in numbers that defy expectation. They are not visible to the eye, nor do they form mats or colonies large enough to shape their surroundings in obvious ways. Instead, they occupy a labyrinth of microhabitats, each cell or small cluster, isolated by meters of rock from its nearest neighbour. Energy here is scarce. There is no sunlight, no rapid turnover of nutrients. Metabolism is slow, sometimes measured in generations per decade. Yet the cells endure drawing energy from the oxidation or reduction of metals from hydrogen, released by the slow reaction of water with iron-rich minerals, or from the breakdown of sulfides. This is life pared to its minimum, sustained by reactions so faint that they barely register as heat. The irony is sharp. Though these organisms live in one of the most stable environments on Earth, their energy supply is so limited that they must remain on the edge of dormancy for most of their existence. Another irony is that despite their isolation, they are part of global cycles. Their waste products eventually re-enter the surface system through groundwater upwelling or volcanic release. A third irony lies in the contrast between their fragility in the lab and their endurance in situ. Many cannot be cultured outside their rock-bound world, yet within it, they have persisted for millions of years. How they came to occupy these depths is still debated. Some researchers propose that they migrated downward from surface or vent environments over geological time carried by infiltrating water. Others suggest they may be relics of ancient microbial communities that formed when the crust itself was young and porous, sealed in as the rock solidified. Modern drilling projects have recovered microbial DNA from depths of several kilometers, and isotopic analyses of trapped gases suggest that some populations have been isolated for tens of millions of years. The cells themselves often appear shrunken with minimal cytoplasm, a possible adaptation to long-term low-energy living. In such conditions, every molecule must be conserved. Repair mechanisms are prioritized over reproduction. Enzymes that mend DNA or restore damaged proteins remain active even when growth has ceased entirely. When small pulses of energy arrive, perhaps from a shift in fluid chemistry, or a fracture that introduces new minerals, the cells rouse, briefly replicate, and then return to dormancy. The rock itself acts as both habitat and archive. Pores are lined with films of clay or quartz surfaces that can absorb and slowly release nutrients. Some minerals provide a direct energy source, acting as electron donors or acceptors in microbial respiration. In basaltic crust, for example, the oxidation of ferrous iron can yield enough energy for minimal metabolic activity. In ultramafic rocks, serpentinization reactions generate hydrogen gas, a valuable fuel for deep-dwelling methanogens. The deep biosphere is a place of continuity. Surface extinctions and climatic upheavals leave it untouched. During global glaciations, when ice covers oceans and continents alike, life in the crust continues much as before. Even asteroid impacts which can sterilize the surface for years fail to reach far into the rock. This stability has led some scientists to speculate that the deep biosphere may represent the largest continuous habitat on the planet, though its biomass is spread so thin that it is difficult to measure. Yet stability does not mean stasis. Over millions of years, geological processes shift the architecture of the subsurface. Faults open and close, aquifers migrate, volcanic intrusions alter temperatures and chemistries. Archaea carried by these slow flows may find new niches or be trapped in pockets that gradually lose their water. In the latter case, some form spores or cyst-like stages awaiting conditions that may never return. Others simply persist in a kind of suspended animation, their metabolism dropping to levels so low they are nearly indistinguishable from non-living matter. Studying such organisms challenges the boundaries of how we define life. At what point does a cell that has not divided for centuries cease to be alive? In the deep rock biosphere, the answer may be less important than the fact that these cells can resume activity when given the slightest opportunity. This persistence has implications beyond Earth. If life can endure in rock-bound water films here, it might also exist beneath the surfaces of Mars Europa or Enceladus, protected from radiation and surface instability. The same adaptation, slow metabolism, tolerance for isolation, use of mineral-derived energy would serve just as well on other worlds. Above these hidden communities, the surface of the planet changes without their knowledge. Continents drift, mountains rise and erode climates warm and cool, yet the deep biosphere remains. The cells here have no sense of time in a human frame. Their only measure is the slow chemistry of their environment, the infrequent arrival of a molecule they can use. If the rock were transparent, one could trace a thin thread of life running through fracture after fracture, down into the mantle's upper reaches, a vast but invisible network, woven into the planet itself. The stillness is not complete. Fluids do move, minerals do react, but it is slow enough to be almost geological. And in this near-geological pace, Archaea have found their place neither in competition with the surface world nor dependent on it, but in quiet parallel to it. Here beneath layers of stone that have never felt the sun, life continues on its own terms, bound to the planet's deep and patient rhythms. Within every cell, survival in the extremes depends not only on the boundary of the membrane, but on the machinery that runs inside it, the enzymes that catalyze reactions, the structural proteins that hold, shape the molecular pumps that maintain gradients. In Archaea, these components are crafted for endurance in ways that differ from all other domains of life. At high temperatures, most proteins unravel their folded shapes, collapsing as heat overcomes the weak forces that hold them together. Archaeal enzymes resist this through tighter packing of their amino acid chains, more ionic bonds linking charged residues, and hydrophobic cores that repel water intrusion. Some even include disulfide bonds, rare in most cytoplasmic proteins, to further lock their shapes in place. The result is a stability that allows metabolic reactions to proceed efficiently in boiling water or near frozen brines alike. There is an irony in this specialization. Enzymes so perfectly tuned to high heat or high salt often fail to work in mild temperate conditions, becoming sluggish or unstable in what most organisms would consider ideal. Another irony lies in their apparent simplicity. Lacking the elaborate compartmentalization of eukaryotes, their cells still perform a full range of life's chemistry, with a minimal set of tools that have outlasted entire geological eras. A third irony is that these proteins designed for stability can sometimes be more flexible in function than those of other life, able to catalyze reactions under a wider range of pressures or pH levels without loss of activity. One enduring mystery is how such molecular designs first arose. Were they the product of direct adaptation to extremes or inherited from ancestors that lived in a far more hostile early Earth? With later lineages losing these traits as conditions softened, comparative genomics shows that many extremophile enzymes share structural motifs absent in bacteria and eukaryotes, suggesting deep evolutionary separation. Yet in some cases, horizontal gene transfer has blurred the picture-moving genes for stability-enhancing proteins between distant lineages. In the lab, scientists isolate these enzymes for use in processes that require heat or chemical resistance DNA polymerases for high-temperature reactions, hydrolases that function in industrial solvents. Such applications are modern, but they echo the same principles that have governed archaeal survival for billions of years. A system is only as durable as its most sensitive component. Supporting the enzymes are small molecules called compatible solutes, which stabilize proteins and nucleic acids without interfering with normal cellular processes. In halophiles, these may be potassium ions or organic molecules like glycine betaine. In thermophils, they may include cyclic compounds that reinforce the hydration shell around macromolecules. These solutes act as molecular cushions, absorbing environmental shocks and smoothing the operation of vital reactions. DNA, too, is protected by specialized proteins that wrap and compact it, shielding it from heat-induced breakage or chemical attack. In hyperthermophils, reverse durase, an enzyme unique to organisms living above about 80 degrees Celsius, introduces positive supercoils into DNA, making it more resistant to denaturation. This adaptation has no counterpart in mesophilic life, underscoring how far the molecular architecture of Archaea can diverge from the norm. Even the ribosomes, the sites of protein synthesis, are adapted for extremes. Archaeal ribosomal RNA includes additional stems and loops that increase thermal stability, and ribosomal proteins carry more salt bridges to hold the structure together under stress. These differences are not superficial. They're woven into every process the cell undertakes, from reading genetic instructions to building the proteins that will maintain its shape and function. Despite these strengths, archaeal molecular machinery is not static. Mutations accumulate over time, and selection continues to refine or alter structures. In some environments, thermophilic lineages have given rise to relatives that prefer cooler temperatures, losing certain stabilizing features in favor of flexibility. In others, halophiles have gained additional pigment systems to expand their light harvesting abilities. This adaptability suggests that even highly specialized enzymes can serve as starting points for new ecological ventures given enough time and the right conditions. Observing these systems in their natural settings reveals the interplay between molecular resilience and environmental challenge. In a vent chimney, for example, a single archaeal species may experience a 50-degree C temperature range across the few millimeters of its biofilm, requiring enzymes that can operate effectively across the entire gradient. In a hypersaline pond, daily evaporation and rainfall can swing salinity from near saturation to levels tolerable by many microbes, so halophils must maintain machinery that works under both extremes, adjusting solute concentrations and enzyme conformations accordingly. The quiet beauty of this machinery lies in its invisibility. No plume or pigment marks the folding of a protein, the binding of a cofactor, the passage of an ion through a channel. Yet these events are the true measure of an organism's capacity to endure. They are also, in a sense, the most ancient echoes of life's origin, for the earliest cells, regardless of their environment, survived only by building molecular structures that could persist in the face of disruption. Over deep time, these adaptations have been tested by planetary change. When the Earth's atmosphere shifted toward oxygen, some archaeal enzymes were rendered obsolete, replaced, or abandoned as oxidative stress reshaped the biosphere. When oceans froze or warmed, when salinity levels rose or fell, the machinery shifted again, shedding parts no longer needed, adding others that gave advantage. What remains today is not a static relic of the early Earth, but a living archive of solutions to problems most life will never face. In the stillness of their environments, whether in boiling springs, hypersaline lagoons, or deep rock aquifers, these molecular systems hum at a slow, steady pace, untroubled by the urgency of faster living species. The cells divide when they can repair when they must, and otherwise wait confident in the integrity of their architecture. The irony, perhaps the deepest of all, is that in building machinery for survival, Archaea have also built tools for persistence, not just over years or centuries, but over spans of time so long that continents have moved and oceans have opened and closed in their tenure. And so at the smallest scale, their enzymes and molecular scaffolds continue their quiet work indifferent to the drama of the surface world, faithful only to the chemistry that keeps them whole. Long before forests shaded the land or reefs shaped the coasts, Archaea were already moving elements through the planet's system, cycling carbon, nitrogen and sulfur, in forms that would later sustain far more complex life. Their role is not dramatic. They do not build visible structures or dominate the biomass of the oceans, yet their influence is planetary. In the earliest oceans without oxygen to drive familiar chemical pathways, archaeal metabolisms filled crucial niches. Methanogens drew carbon dioxide from the water and combined it with hydrogen to form methane, releasing it into the atmosphere where it trapped heat from the faint young sun. Sulfur metabolizing archaea converted elemental sulfur to hydrogen sulfide or oxidized sulfides to sulfuric acid, linking volcanic outgassing to ocean chemistry. Ammonia oxidizing archaea transformed reduced nitrogen compounds into nitrate, making them available to other microorganisms in a form that could support growth. There is an irony in this quiet productivity. While the rise of oxygen-producing cyanobacteria would later dominate the narrative of Earth's transformation, it was these anaerobic archaea that stabilized the early biosphere, smoothing chemical fluctuations that could otherwise have destabilized climate or nutrient availability. Another irony is that their cycles often operated invisibly, buried in sediments or deep water, where their contributions left only chemical fingerprints in ancient rock. A third irony lies in how dependent modern ecosystems remain on them, despite their invisibility in certain habitats today, such as the open ocean's mesopelagic zone. Ammonia oxidizing archaea are among the most abundant organisms on Earth. The origin of these cycles is still under study. Genetic evidence suggests that many archaeal metabolic pathways diverged early from bacterial equivalents, possibly representing independent solutions to the same geochemical opportunities. In some cases, their enzymes share no common ancestry with those of bacteria performing similar reactions, hinting at deep evolutionary experimentation in the planet's first billion years. The persistence of these pathways through shifts in climate-ocean chemistry and atmospheric composition suggests they are among life's most robust designs. In modern hot springs, deep-sea sediments and hypersaline lakes, archaeal nutrient cycles still operate in forms close to their ancient patterns. Methanogenesis continues in anoxic muds, producing a gas that bubbles to the surface and escapes to the air. Ammonia oxidation still occurs in the twilight waters of the open ocean, its product feeding phytoplankton blooms that in turn sustain food webs reaching to fish, seabirds and whales. Sulfur cycling persists in volcanic soils and hydrothermal vents, binding elemental chemistry to the rhythm of life. The scale of their impact is disproportionate to their size. In global climate history, for example, methane from archaeal methanogens may have been a key factor in preventing the planet from freezing entirely during periods when the sun's output was significantly lower than today. In the geological record, isotope ratios in carbon-bearing minerals suggest large and sustained inputs of methane in the Archean and Proterozoic eons. Later during snowball Earth, glaciation's methane production from subglacial or hydrothermal habitats may have helped thaw the planet. In nitrogen cycling, ammonia oxidizing archaea regulate the availability of nitrate in the ocean, influencing the productivity of phytoplankton, and thus the drawdown of atmospheric carbon dioxide. These cycles reveal another quiet truth even in the absence of oxygen. Life can weave itself into planetary feedbacks shaping climate and chemistry, without the structures or behaviors we often associate with ecological engineering. The adaptability of these cycles is striking. In oxygenated waters, ammonia oxidizing archaea operate efficiently at extremely low ammonia concentrations, outcompeting bacteria for scarce resources. In high salt environments, methanogens maintain their pathways despite osmotic stress by tuning enzyme structures and internal solute balances. Sulfur metabolizers shift between reduction and oxidation, depending on which electron donors or acceptors are available, maintaining a steady throughput of energy regardless of external fluctuations. These flexibilities have likely been key to their survival through mass extinctions and climate upheavals. They have no need to migrate or escape. They alter their metabolism in place, matching their pace to the availability of substrates. The mystery is how early in Earth's history such complex cycling emerged. Did Archaea inherit core pathways from a last universal common ancestor, or did they innovate independently after splitting from the bacterial line? The fossil record offers little clarity, as the soft-bodied microscopic forms of Archaea leave almost no direct trace. Instead, geochemists rely on isotopic patterns, mineral associations, and rare chemical fossils such as archaeal lipids to reconstruct their presence. These traces, while sparse, suggest that archaeal nutrient cycling was well established by at least 3 billion years ago. Observing them today, one might stand on the shore of a hypersaline lagoon, see only a flat expanse of water and salt, and never suspect that within its sediments, Archaea are shuttling nitrogen, sulfur, and carbon between forms influencing gas exchange with the atmosphere. Or one might hover over a deep-sea vent, watch plumes of mineral-rich water rise into the dark, and not see the invisible work of Archaea, linking the chemistry of the mantle to the surface oceans. It is a reminder that planetary systems are not always driven by the largest or most visible actors. Often they depend on the smallest, those whose presence is revealed only through the patient language of chemistry. Over time, as oxygen spread and other life forms evolved to occupy the upper layers of the ocean, and the land Archaea did not vanish. Instead, they retreated into niches where their ancient cycles could continue undisturbed: anoxic sediments, deep subsurface aquifers, extreme salinity pools, acidic springs. In these refuges, their processes run as steadily as they did when the continents were bare rock and the air was heavy with volcanic gases. The irony is that while humans often imagine Earth's biosphere as dominated by visible plants and animals, its stability rests in no small part on these hidden chemists, whose metabolisms have kept the planet's systems in balance across eras when the surface was anything but stable. In this way, Archaea remain not only survivors, but active shapers of the world, their ancient cycles still turning beneath our feet and far beyond our sight, under a sky dimmer than today's, when the young sun gave only a fraction of its later warmth. Methane from Archaea may have been the thin blanket that kept Earth's surface from freezing solid. In the anoxic oceans of that time, methanogens flourished in sediments, hydrothermal vents, and subglacial pockets, drawing carbon dioxide from the water and combining it with hydrogen to exhale methane. The gas drifted upward, accumulating in the atmosphere in concentrations far higher than any known today, trapping infrared radiation and warming a planet lit by a weaker sun. The irony is that the very gas now associated with human-driven climate change may once have been the difference between a habitable Earth and a frozen, lifeless world. Another irony is that methanogenesis is at its core a simple process, a handful of reactions powered by a small suite of enzymes, yet it exerts planetary-scale influence. A third irony lies in how poorly the geological record can pin down its magnitude. Methane is invisible in ancient air bubbles, its presence inferred instead from isotopic shifts in carbon-bearing rocks that are open to more than one interpretation. Methanogens are strict anaerobes, Oxygen is toxic to them, disrupting their enzymes and damaging their delicate cofactor assemblies. In the early Earth's oceans, this was no obstacle. Oxygen was absent and hydrogen and carbon dioxide were abundant. Even after the Great Oxidation event, when oxygen began to seep into surface waters, methanogens retreated into sediments, wetlands, and other anoxic refuges, their metabolism unchanged. They survive today in rice paddies, ruminant guts, and Arctic permafrost, still producing methane as they have for billions of years. Their enzymes, such as methyl coenzyme M. reductus, are tuned to the precise chemistry of this pathway, binding nickel-based cofactors that facilitate the final step in methane formation. These proteins are ancient, conserved across diverse methanogen lineages, suggesting they arose early and have been maintained with little change. The role of methane in Earth's deep history remains a subject of active study. Models of the Archean atmosphere suggest that methane could have reached levels hundreds of times higher than present, potentially forming a photochemical haze that altered the planet's climate and surface radiation environment. Such a haze, while reducing some sunlight, could have further trapped heat and shielded surface life from ultraviolet radiation, creating a feedback loop favourable to early microbial communities. Yet the stability of this methane-rich atmosphere would have been fragile. A surge in oxygen production from cyanobacteria could oxidize methane into carbon dioxide, weakening the greenhouse effect and triggering global glaciation. Some scientists link the Paleoproterozoic snowball Earth events to such a collapse, when rising oxygen reduced methane to levels too low to offset the sun's faintness. In this way, methanogens may have been both guardians and victims of Earth's early habitability. Their decline in open oceans did not end their influence. In wetlands and anaerobic sediments, they continued to contribute to atmospheric methane, albeit at lower concentrations. In cold climates, they adapted to life beneath ice or in permafrost, where trapped organic matter provides a slow but steady supply of substrate. In the deep biosphere, they use hydrogen from water rock reactions, producing methane that can remain trapped for millennia in hydrate form beneath the seafloor. Modern climate change has brought their role back into focus. Warming permafrost releases both organic matter and methane hydrates, feeding methanogens and potentially increasing methane emissions. The same gas that once preserved Earth's warmth now risks amplifying it beyond stability, a reversal of fortune written in planetary chemistry. The mystery of methanogen origins ties into broader questions about life's early evolution. Did methanogenesis emerge in hydrothermal systems where hydrogen and carbon dioxide were naturally abundant, or did it arise in sedimentary environments, perhaps linked to fermentation pathways from other anaerobes? The isotopic signature of methane in ancient rocks strongly depleted in carbon-13 hints at biological origins, but abiotic methane from serpentinization can produce similar signals, making absolute certainty elusive. Modern analogues such as deep-sea vents and hypersaline lagoons provide partial clues. In some hydrothermal systems, methanogens live alongside other extremophiles, exchanging metabolites in tightly knit consortia. In others, they are the primary producers, feeding higher microbes in the absence of photosynthesis. The genetic diversity of methanogens today suggests they radiated into multiple lineages, early colonizing a range of anaerobic niches long before complex life appeared. Standing at the edge of a present-day methane seep, one might see only bubbles rising from the sediment, each one carrying gas from microbial processes deep below. No plume marks their origin, no colour betrays their presence, yet their chemical imprint can stretch across ecosystems and influence climate far beyond the boundaries of their habitat. In the frozen soils of the Arctic, they wait in dormancy through long winters, reactivating in the thaw to release pulses of methane into the air. In the guts of grazing animals, they transform the hydrogen produced by fermentation into methane, which escapes in every exhalation. On the ocean floor, they feed slow leaks of methane into currents that may carry it to the surface. The quiet consistency of their work is both their strength and their danger. They are indifferent to whether the planet is in balance or crisis. Their metabolism has no setting for caution. In the scale of planetary history, methanogens are reminders that life's chemistry can be both stabilizing and destabilizing depending on context. The same reactions that once held Earth above freezing now add to the accumulation of greenhouse gases in an already warming atmosphere. This dual role is not a contradiction, but an outcome of life's adaptability. It is the planet's changing conditions that turn benefit into risk. And so methanogens continue as they have for billions of years in the darkness of sediments, in the chill of permafrost, in the mineral warmth of vents, untroubled by the shifting meanings their methane holds for the world above. Life in extremes is not simply a matter of surviving the moment. It is the art of persisting through deep time, carrying the means to endure conditions that shift on scales of centuries, millennia, or entire geological periods. For Archaea, this endurance rests heavily on the architecture of their genetic systems, the ways in which DNA is preserved, repaired, and passed on. In most organisms, DNA is a vulnerable molecule prone to damage from heat radiation, chemical attack, and the inevitable errors of replication. In the environments Archaea inhabit, boiling vents, hypersaline lagoons, acidic pools, deep rock fractures, the potential for such damage is constant. Yet their genomes remain stable enough to bridge uncounted generations. Part of this stability comes from proteins that bind DNA, wrapping it into compact structures that shield it from physical and chemical harm. Some archaeal DNA-binding proteins resemble histones, the same type found in eukaryotes, hinting at a deep evolutionary connection. Others are unique to Archaea, tailored to their extreme niches, with surfaces that repel reactive chemicals or fit tightly around DNA to exclude damaging agents. The irony is that in these protective configurations, DNA is less accessible, potentially slowing processes like transcription, yet the trade-off favors survival over speed. Another irony is that many Archaea, despite inhabiting environments where life is sparse and competition minimal, invest heavily in repair mechanisms usually associated with organisms under intense evolutionary pressure. A third irony lies in the nature of their mutation rates. They are neither as low as might be expected for such stability, nor so high as to threaten long-term viability, suggesting a balance that permits slow adaptation without risking collapse. Their repair systems are varied and often more versatile than those of other prokaryotes. Base excision repair corrects small chemical modifications. Nucleotide excision repair removes bulky lesions. Homologous recombination patches double-strand breaks with precision. In hyperthermophiles, enzymes operate at high temperatures without losing activity, relying on structural features that maintain folding under thermal stress. Some Archaea possess reverse gyrase, the enzyme that positively supercoils DNA, adding an extra layer of thermal stability. This enzyme is absent from mesophils and is considered a signature of life at very high temperatures. In the deep biosphere, where cell division is rare, the emphasis shifts from replication fidelity to long-term preservation. Cells may go decades between replications, relying on constant surveillance and repair of their genetic material to prevent cumulative damage. In such conditions, the energy cost of repair is far less than that of replacing a lost lineage through reproduction. Horizontal gene transfer also plays a role even in extreme environments where cell density is low. Mobile genetic elements, viruses and plasmids move between Archaea, bringing in new genes or alternate versions of existing ones. Some of these elements may be ancient, persisting in a host-switching cycle for hundreds of millions of years. The presence of shared metabolic genes across distant archaeal lineages suggests that such exchanges have helped spread adaptive traits, allowing communities to respond to environmental changes without the slow wait for beneficial mutations. The preservation of functional genes over immense timescales points to another quiet mystery. How do these lineages avoid the gradual degradation predicted by genetic drift in small populations? One possibility is that even rare selection events, a sudden chemical shift, a change in temperature can purge less fit variants, restoring genomic integrity. Another is that the interplay between dormancy and occasional bursts of activity resets the evolutionary clock in ways we do not yet fully understand. Modern studies in hot spring salt lakes and subsurface aquifers show that many archaeal genomes are surprisingly compact. Lacking the redundancy common in bacterial genomes of similar size, this efficiency may be a product of long adaptation to resource scarcity, where maintaining only essential genes reduces the metabolic cost of replication and repair. Yet within these streamlined genomes are often clusters of genes for uncharacterized proteins, many of which may be involved in adaptations we have yet to recognize. Some might mediate interactions with minerals, others might control the uptake or export of solutes in ways specific to extreme chemistries. The slow pace of archaeal evolution compared to that of many bacteria gives them a kind of geological patience. In an environment that remains chemically and physically stable for millions of years, there is no advantage in rapid change. Instead, stability is preserved, ensuring that the molecular systems proven to work are not disrupted by unnecessary novelty. This constancy is not the absence of evolution, but its refinement toward a narrow set of solutions. The irony is that such refinement can make lineages highly specialized, yet in extremes, specialization often overlaps with general resilience. A membrane that resists acid will also resist many solvents. An enzyme that works in boiling water will function well in moderate heat. The genetic architecture that encodes these traits thus acts as both a key and a lock, preserving the ability to live in the narrowest of niches, while incidentally enabling survival in others. Observing this stability over deep time reframes our sense of what adaptation means. In the human view, adaptation is often tied to visible change to species shifting form or behavior over generations. For Archaea, adaptation may mean holding a form steady for hundreds of millions of years, allowing the environment to fluctuate around it. The blueprint is not altered unless absolutely necessary, and when it is, the changes are often small, a modification to an enzyme's active site, an alteration in the lipid composition of a membrane, a shift in gene regulation that changes the timing of a response. In every case, the core remains intact. From the perspective of planetary history, this genetic resilience is one of the reasons Archaea have been such constant players in Earth systems. Their metabolic pathways, encoded in durable genomes and protected by efficient repair systems, have operated without interruption across eons, bridging global transitions that erased entire lineages of more complex life. In the heat of vents, in the stillness of salt crusts, in the shadows of rock pores kilometers deep, their DNA remains not unchanged, but enduring a record of adaptation, written in sequences that have faced the full spectrum of the planet's extremes and continued on. Beyond their role on Earth, Archaea offer a living framework for imagining life elsewhere. The environments they inhabit, boiling vents, hypersaline basins, acidic lakes, subglacial aquifers, are analogues for conditions found or suspected on other worlds. Their resilience makes them models for astrobiology, where the search for life is shaped by the recognition that habitability is not limited to mild climates or abundant sunlight. On Mars, salt deposits suggest that hypersaline brines once existed, and perhaps still do beneath the surface. Halophilic archaea on Earth demonstrate that such brines can host active life even when they are nearly saturated with salt-drawing energy, from light through bacteriohodopsin, or from slow chemical reactions in the absence of oxygen. The irony is that these earthbound microbes may be more similar to potential Martian life than to most organisms we encounter in daily experience. Another irony is that while the popular image of alien life tends toward the complex or exotic, the most probable form may be microbial, simple in appearance, but extraordinary in endurance. A third irony lies in the fact that the same traits that allow Archaea to thrive in extremes — slow metabolism, tolerance for desiccation, resistance to radiation — also make them difficult to detect, as their activity leaves only faint chemical traces over long spans of time. Europa and Enceladus, icy moons of Jupiter and Saturn, harbour subsurface oceans warmed by tidal flexing. Hydrothermal activity at their seafloors is a strong possibility inferred from plumes detected in space. On Earth, hydrothermal vents are among the richest archaeal habitats supporting methanogens and sulfur metabolizers that require no sunlight. If similar vents exist beneath the ice of these moons, Archaea-like life could persist there indefinitely, recycling nutrients within isolated ecosystems. The challenge for exploration is that such life, even if abundant locally, might be nearly invisible when sampled from plumes diluted in vast oceans. In the lab, researchers simulate extraterrestrial conditions to test archaeal limits, exposing cells to intense radiation, repeated freeze-thaw cycles, or extreme vacuum. Many survive these trials, especially those capable of forming protective states or embedded in mineral matrices. In salt crystals, halophils have been shown to remain viable for years, with no metabolic activity reactivating when rehydrated. This persistence in salt, mirrored in evaporate deposits on Mars and possibly elsewhere, raises the possibility that life could survive long periods of planetary dormancy, waiting for transient habitable conditions. Another parallel lies in subglacial environments on Earth, where lakes beneath kilometers of ice remain liquid due to geothermal heat and pressure. In such settings, chemolithotrophic archaea oxidize minerals or reduce sulfur compounds to gain energy, operating entirely without sunlight. Similar niches could exist beneath the ice caps of Mars or within the thick ice shells of outer solar system moons, where isolated liquid water and rock interact over geological timescales. The mystery is whether life in such places would resemble Earth's Archaea due to convergent adaptation, or whether entirely different molecular solutions could arise. Our understanding of life's boundaries comes largely from studying extremophiles like Archaea, yet these boundaries continue to expand as new environments are explored. The concept of a habitable zone has shifted from being defined purely by temperature and distance from a star to one that includes any setting with liquid solvents, energy gradients, and chemical building blocks, all of which Archaea exploit in varied and subtle ways. Their metabolic diversity suggests that extraterrestrial life might not rely on the same energy sources as surface life on Earth. Methanogens, for example, require only hydrogen and carbon dioxide, both common in planetary and moon interiors. Sulfur metabolizers could thrive in volcanic settings, while halophiles could persist in brines beneath evaporitic crusts. Even acidophiles could find analogues in sulfuric acid-rich environments like those observed on Io, or hypothesized in certain exoplanetary atmospheres. The irony in this astrobiological perspective is that the most alien-seeming forms of life may be those already living quietly on our own planet, largely ignored outside scientific study. Another irony is that by learning to detect Archaea in Earth's harshest niches, we're also developing the tools to find life elsewhere, yet the faintness of their chemical signatures means we may overlook them, even if they are present. Space missions now aim to target environments most analogous to archaeal habitats, the planned sampling of Mars's subsurface ice, the analysis of plumes from Europa and Enceladus, the study of Titan's methane lakes. Each carries the hope of finding biosignatures, isotopic ratios shifted by biological activity, complex organic molecules arranged in non-random patterns, or microscopic structures consistent with cells. The interpretation of such data will lean heavily on what we know from extremophiles on Earth, especially the ability of Archaea to produce distinct chemical markers under extreme conditions. If such markers are found, the question will not only be whether they indicate life, but whether that life shares ancestry with Earth's, perhaps transferred through meteorite exchange, or whether it arose independently. In both cases, Archaea provide a reference point, an example of how life can be both minimal in structure and maximal in resilience. Even without direct discovery, studying Archaea reshapes our expectations of habitability. They show that liquid water need not be abundant, that energy can be harvested from chemical gradients invisible to the naked eye, and that survival is possible under pressures, temperatures, and chemistries once considered sterile. They remind us that adaptability is not about thriving everywhere, but about finding and holding a stable place in the vast range of planetary conditions. In this sense, Archaea are not just models for extraterrestrial life, but also for the persistence of life itself. They bridge the gap between the earliest biochemistry on Earth and the potential for life far beyond it, offering clues not only to what might be found, but to how we might recognize it. The search for life elsewhere is in part the search for what Archaea have already proven, that the boundary between the possible and the actual is wider and more quietly inhabited than we once believed. Across the planet, from the searing outflows of volcanic springs to the lightless pores of ancient rock, there are lineages of Archaea whose ancestry stretches back billions of years with little visible change. These are the time's invisible colonies communities that persist not through expansion or conquest, but through remaining in place, holding their ground against the slow churn of geology and climate. Many occupy refuges so stable that evolutionary pressure for novelty is minimal. A halophile in a salt-saturated lagoon today may be nearly indistinguishable from its counterpart in a Proterozoic evaporate, its membrane lipids, pigment systems, and core metabolic pathways essentially unchanged. The irony is that in a biosphere where adaptation is often equated with change, these organisms survive precisely because they do not change, embodying a strategy of conservation rather than innovation. Another irony is that their habitats, though geographically scattered and chemically distinct, can host Archaea whose genetic differences are surprisingly small. Salt lakes separated by continents may harbour near-identical strains, dispersed by wind-blown crystals or migratory birds over spans of millennia. A third irony lies in the invisibility of their persistence. They are not part of the visible world of forests, reefs, or herds, yet their quiet endurance outlasts many of those more conspicuous forms. The stability of these colonies depends on the constancy of extreme conditions. In deep subsurface aquifers, the chemistry of water-rock interactions changes only over geological timescales, giving resident Archaea an unhurried pace of life. In hydrothermal systems, even though individual vents may be short-lived, the global network of tectonic ridges ensures a continuous supply of similar habitats. In hypersaline basins, evaporation and inflow reach a balance that maintains high salinity year after year, century after century. These conditions favor lineages that can resist occasional disruptions, a fault shift, a seasonal flood, a temperature anomaly, but otherwise remain in equilibrium. Genetic studies reveal that some of these populations have extremely low mutation rates, a possible consequence of both their slow growth and robust repair systems. This genomic conservatism means that when environmental conditions do shift beyond their tolerance, extinction is more likely than adaptation. Yet this risk is balanced by the rarity of such shifts in their chosen refuges. Over deep time, the strategy succeeds. Extinction may claim individual colonies, but the lineage as a whole persists by recolonizing newly formed habitats that match their narrow requirements. The mystery is how these ancient lineages weathered global upheavals that transformed most of Earth's surface. During mass extinctions, snowball glaciations, and prolonged volcanic winters, surface ecosystems were repeatedly disrupted, but the extremes that Archaea favour often remained intact. Beneath kilometres of ice brine pockets and geothermal vents continued to operate. Deep beneath the seabed, hydrothermal activity went on largely unaffected by surface climate. Even in global freeze scenarios, salt-rich waters beneath ice could maintain liquid phases where halophils endured. These hidden havens acted as time capsules sheltering communities that would later recede the extremes once conditions elsewhere stabilized. In the modern world, these colonies are still easy to overlook. A salt flat might appear lifeless to the casual observer, yet beneath its surface halophilic mats and biofilms line brine channels. A barren volcanic slope may hide acidophiles within its steaming fissures. A deep borehole may bring up water clouded with cells from a biosphere that has never seen sunlight. Their distribution is patchy, yet their persistence gives them a cumulative presence across the planet's most inhospitable terrains. There is a quiet efficiency in their way of life. With no need to invest in rapid reproduction, elaborate structures, or competitive strategies, their energy budgets are focused on maintenance, preserving membrane integrity, repairing DNA, stabilizing proteins. The balance is delicate but effective, allowing them to wait out unfavorable fluctuations and resume full activity when conditions return to optimal. This patience makes them resistant not only to natural disturbances, but also to human attempts at eradication. In saline industrial waste ponds, acid mine drainage, or geothermal energy wells, Archaea frequently appear without invitation, drawn by the chemical parallels to their natural homes. These anthropogenic extremes act as modern analogues to ancient habitats, and Archaea move into them readily, sometimes dominating microbial communities within months. The global spread of such environments has in some cases expanded their range, offering new refuges even as other habitats shrink. Still, their long-term survival depends on conditions humans cannot easily replicate: the slow, stable rhythms of geology. Artificial extremes may come and go too quickly, disrupted by remediation or changing industrial practices to serve as permanent colonies. The irony is that the most stable environments for these organisms are often the least accessible to study deep within the crust under polar ice or in remote volcanic fields. Sampling them requires specialized drilling submersibles or long expeditions to isolated terrain. As a result, our picture of their diversity is incomplete based on the few sites we have been able to explore. Each new discovery often adds unexpected branches to the archaeal tree of life, suggesting that the true scope of their persistence is far greater than current records show. From a planetary perspective, these invisible colonies represent continuity across the vast discontinuities of Earth's history. They are threads woven through every chapter, linking the chemical landscapes of the Archean to the present day. They have no interest in the surface dramas of evolution, no stake in the rise and fall of dominant species. Their survival is not about thriving in the moment, but about outlasting the moment altogether. In their stillness, they remind us that change is not the only path through time. Sometimes the surest way forward is simply to remain. Beneath storms that scour the surface, beneath seasons and epochs, there is a quieter layer of the planet where life is untouched by weather or tide. Here in heat, salt acid and stone Archaea remain as they have for billions of years, their rhythms matched not to the turning of the earth or the pull of the moon, but to the slow pulse of geological time. These are the silences beneath all storms' refuges, where energy flows steadily, where chemistry is constant enough for entire lineages to persist without alteration, and where extinction is an event measured in continents shifting rather than decades passing. The irony is that the most enduring life on the planet is also the least visible. A rainforest canopy changes with every season. Coral reefs grow and erode over centuries. Yet in a deep rock aquifer, the same lineage of Archaea can hold its niche for millions of years, changing less in that span than a forest might in a single year. Another irony is that the very conditions that would destroy most life heat, that warps protein salinity, that collapses cells acidity, that strips away structure, create the stability that Archaea depend on. By choosing the extremes, they avoid the instability of the moderate, where competition, predation, and environmental fluctuation are constant. A third irony lies in the fact that while their individual cells live brief spans, dividing and replacing themselves, the genetic identity of the lineage is preserved so faithfully that it functions as an unbroken presence in the planetary record. These environments are immune to the cycles that dominate the biosphere above. A volcanic vent may collapse, but another will open nearby, fed by the same mantle heat. A salt flat may dry, but the next season's flood will restore the brine. Even the planet's great upheavals, meteor impacts, glaciation, shifts in atmospheric composition leave many of these refugees untouched. It is in these unchanging places that the quietest revolutions in life's history have unfolded, not in bursts of innovation, but in the perfecting of endurance. Scientists searching for the earliest traces of life often find their best analogues here. The isotopic fingerprints of ancient methane, the structure of preserved lipid molecules, the mineral patterns formed by sulfur metabolism all have living counterparts in modern Archaea inhabiting the same kinds of extreme environments. Studying these organisms is like looking into a mirror that reflects not just the present, but deep time itself. Their continued presence challenges the idea that evolution is always a march toward complexity. For Archaea in these extremes, complexity in form is unnecessary. Complexity in chemistry is everything. The molecular machinery that keeps them alive, the enzymes, the membrane lipids, the repair systems has been honed to perfection for a particular set of conditions. Alter the conditions and the system fails. Keep them stable, and it runs indefinitely. This dependence on environmental constancy makes their persistence even more remarkable, for it means that the planet has for billions of years maintained pockets of stability despite surface chaos. The stillness here is not absence, but balance. In a vent chimney, minerals precipitate at the exact rate needed to keep channels open for hot chemical-rich water to flow. In a salt-saturated lagoon, evaporation and inflow remain in rough equilibrium, preventing the dilution that would drive halophils out. In deep crustal pores, the slow seep of hydrogen from water-rock reactions is matched by the equally slow uptake of Archaea, using it for metabolism. These are ecosystems without seasons, without sudden shifts, where the measure of time is the accumulation of mineral layers, or the drift of continents. And yet nothing is truly permanent. Vents collapse, lakes shift, rock fractures close. When that happens, Archaea persist not by adapting to the change, but by moving carried in currents in wind-blown crystals, in migrating sediments until they find another refuge with familiar chemistry. The lineage survives because somewhere on the planet the same conditions exist again. The mystery is how many of these refugees we have yet to find. Our exploration of extreme environments is limited, and each new deep drilling project, each remotely operated submersible, reveals communities where we had assumed there was only mineral and water. Genetic sequencing from such sites often returns lineages with no close matches in existing databases, suggesting that much of archaeal diversity is still hidden, not in unexplored species. Rich rainforests, but in quiet elemental pockets beneath our feet and oceans. Their persistence across geological time raises questions about life's potential future. Long after forests and reefs have shifted or vanished after climates have warmed or cooled beyond current bounds, it is likely that Archaea will remain in their extremes largely untouched by changes above. They are life's baseline, the enduring layer that continues when the more visible layers fade. In this way, they connect the Earth of today to the Earth of its earliest days, and will likely connect it to the Earth of its distant future. Standing at the edge of a salt flat at dusk, or at the rim of a steaming vent field, it is difficult to see this continuity. The air is still the ground, bare the water opaque. Yet beneath the surface cells divide, enzymes fold, gradients are maintained, the same processes that have unfolded in these conditions for billions of years. The surface may change, the sky may shift from blue to red over geological spans, but here the work continues without pause. In these silences beneath all storms, the Archaea endure not as relics, but as active participants in the planet's ongoing chemistry. They are proof that survival does not always belong to the swift or the adaptable in the usual sense, but to those who match their pace to the deepest rhythms of the Earth, and who hold their place until the planet itself moves beneath them. Now the journey slows. The vents cool in the mind's eye, their mineral plumes dissolving into still water. Salt flats fade under a low horizon, their mirrored surfaces dimming with the last light. The hiss of acid pools softens to a faint breath, and the deep rock aquifers grow quieter still, their hidden waters unmoving. The images of Archaea in heat, in salt, in stone drift together into a single steady presence. They are no longer in separate habitats, but part of a continuous layer beneath the surface, a low hum of chemistry that has never stopped. There is no urgency here, no seasons to keep, no predators to watch, for no tides to answer. Only the slow exchange of molecules, the rise and fall of gradients, the quiet adjustment to whatever changes come at a pace the cell can match. The surface may hold storms, but down here storms are only distant echoes. In this stillness, time stretches. A single division might span weeks. A lineage might span continents without ever hurrying. The patience of it seeps in until the boundaries between years and centuries lose meaning. The mind too can loosen its grip on the clock, settling into that slower measure. The planet holds these refugees like embers in the dark, each one small and self-contained, yet together spanning the crust and oceans. They have been there since before the air held oxygen, before the continents had their current names, before forests or animals drew breath, and they will remain quietly when the shape of the land and the temperature of the air have changed again. The last light fades from the salt. The vent's plume is only a shadow now, its minerals falling back to the seafloor. In the rock pores, cells settle into their steady work unchanged by night. The surface above sleeps, and in these silences, beneath all storms, so does the thought of them. The mind follows that quiet into its own and rests.